I forgot to mute. Good morning, everyone. Can you hear me?
I can hear you.
Okay. Great. Can you see the slides?
Yes.
Great, thank you. So good morning, everyone, and thank you so much for the invitation. What I'm going to talk about this morning is our work on designing this simple test and rapid testing for food monitoring. I am going to present the work of maybe 4 or 5 of my graduate students here in the picture. I will start by saying that I'm not a food expert, but we are doing a lot of sensing work, and what we do is driven primarily by the excitement of scientific discovery. So I have an issue with the computer. I cannot move the slide. So what I will try to do is I will leave it like that without the landscape. So the outline, first, I will go over some of the materials and sensors, the concept design and applications for those of you who are less familiar with sensing technology. And then I will show you a couple of examples of low cost that we designed in our lab for food analysis, so particularly with the idea of implementing this type of sensors for packaging. Some of the sensors that we've done are targeting hypoxanthine lactate, ethanol, bisphenol A, and heavy metals. And I'll also talk about manufacturing and integration of these devices into the food processing. So if you think of sensors and material solutions in a sustainability context, what sensors could give is on-site measurements, affordable or accessible response. So we are looking for something that is deployable, scalable, and low cost. For conventional testing and conventional analytical technology like mass spectrometry or chromatography, typical analysis involve collection of sample processes, perform the test, and then compile the results and analyze the results. If we look at a simple solution and a field deployable sensor, ideally we will either load the sample or the sensor will be directly incorporated within the sample, and it will provide an immediate results. And this can provide significant economic and social benefits. If we look at the sensing technology and the success stories really in the sensing and biosensing field, the well-known glucose meter is the prime example that uses an enzyme, glucose oxidase or glucose dehydrogenase, the lateral flow strip, pregnancy test strip, and we've seen lately with the COVID pandemics, the inexpensive, easy-to-use COVID test. And there is a lot of work going on in the sensing field and field deployable and rapid test, lateral flow devices, paper-based devices, lab on a chip, a lot of work in wearables and these sensors on flexible and conductive substrates. However, I think that the implementation of the sensing, of this smart sensing in food is somehow behind. So how and what are the challenges and why is this technology, how can we translate this technology from the benchtop into the field? If we think of the food analysis itself, and we try to push this idea of incorporating or developing sensors that could go into food packaging, that can provide a direct real-time information on the quality and safety of food at the time of use. So we can envision a label that is directly added to the package and it changes the color as food starts to degrade, or an entire package that changes appearance of food as food degrades, so color changing films, or a system that can detect contamination as we place it inside or in contact with food. Ideally, we would like this to be to not require any sample prep, which is extremely difficult to implement and maybe not possible with some solid matrices. So what will these sensors do is to improve the communication with the consumers, provide an indication of the safety. We can also think of adding to this smart packaging a variety of other functions, for example, antimicrobials, like antibiotics or antimicrobial peptides, temperature regulation for oxygen scavengers, materials that can release compounds, freshener enhancement, flavors or antioxidants, or materials that can retain compounds that are responsible for degradation, such as ethylene, or oxygen, UV light, moisture, and humidity. So we try to look at this idea of smart sensors and how and what are the possibility of sensing technology and what they will do. First, it's possible to reduce the food waste and extend the lifetime of food. This is an example I found for food waste in Europe, in European country, and about 40% of the food that is consumed globally, it becomes waste. So can we extend the food by using that packaging, and can we also provide indicators that will will be used in lieu of date, sale by date or due date, and relate the food composition with the actual content, the label with the actual content. So if we think of sensing technology, one can measure gas evolution, for example, ethylene for fruit ripening, or volatile amines measured as a pH change on a pH type paper. We can think of toxins, allergens, or meat products of ATP degradation that could be either emitting, absorbing, in addition to measuring the food. So, and if we look in the literature, there are a variety of sensing that have been developed for pathogens, for adulterants, for degradation metabolites, allergen, toxin. But if we look at the current state of the art, most of the sensors are tested on standard samples, very few in actual food samples, and almost none in actual packaging. So we have a lot of fundamental research going on, but somehow these beautiful sensors in hundreds of publications do not make it into even real testing or consumers use or industry used. And some of the reason for this and why is most of the sensors are sophisticated, they require multiple steps, they are sensitive, especially biological sensors that incorporate enzymes, antibodies, they degrade fairly easy, and they don't withstand the conditions of actual food. And what are the challenges? It's really high cost of existing technology, lack of long-term stability, sensitivity and selectivity is one thing when we do measurement on standard solutions in the lab. It's another thing when we do it on food samples. Multi-step procedures and expensive reagents. I will also list as challenging standardization and FDA approval, as well as regulatory map. Many sensors developed in the last 10 years have a lot of nanotech products incorporated within, and there is a whole debate as to whether these nanotech products are posing any toxic effects if we are to use them in consumer products or packaging, especially related to food. So human exposure to nanomaterials become an issue. If we think of ideal requirement and what will this sensor look like, self-powered ideally, equipment-free. So we can look at the color change or an RFID or something that will transmit the signal to a cell phone. Reagentless is not that the sensor by itself is reagentless, but all the reagents should be incorporated within the same structure, so that we can do one-step analysis and not multi-step analysis. Easy data interpretation and interface with consumer is also very important. Stable and operational in real world conditions. So if we are looking at field supermarkets or at home, and biocompatibility. So FDA approved materials, it's important, and not and also large-scale production, something that is manufacturable and scalable. If we want to add something to packaging, we are looking at millions of billions of packaging, so we cannot add $1 a piece. We are looking at cents values for one of these sensors. And many of us who are doing sensors and biosensors in the lab, we know that we are far from that value. And potentially adaptable to packaging. Packaging is one example, but it can be used for point of harvest and for processing facilities, as it was discussed in the prior talk. So with that in mind, we started to work on this sort of devices about 10 years ago with the idea, can we convert this mass spectrometry or HPLC into something that is extremely inexpensive and mass-produced, selective, simple to use, sensitive, and that we could potentially integrate into packaging. So we need an all-in-one device with built-in recognition and detection capabilities. And this picture here are examples of food labels that we have developed in the lab. So what I will do in the next few slides is to show you some examples of platforms that we have developed. Most of them are on paper and plastic sensing support. So we spend quite a bit of time of modifying paper or plastic or PET and impart functionality to these surfaces. Can it be used as a multifunctional device? Can it be mass produced? Can we put active bioreagents? For example, enzymes and signal responsive materials that will provide a direct optical or electrical response for detection. Can we get selectivity from these materials? So we become very experts in modifying paper, cotton, we started to use organic dyes. However, we very easily noticed that those degrade over time, and it's quite difficult to, and they leach over time. So we replaced all of the organic dyes with inorganic nanoparticles or redox nanoparticles that are changing colors and they are preserved in immobilized states. We also develop procedures to immobilize enzymes. So we basically silanize the paper, and then we are building layers. We use layer by layer deposition of the bioreagents. Most of the work that we do is with enzymes and biopolymers such as chitosan and alginate. We try to use chemical deposition or chemical functionalization with glutaraldehyde or EDC chemistry. Most of those degraded much faster, and the activity of the bioreagents were significantly reduced. The next step was to mass produce. So every single structure that we produce, we try to print. We've done a lot of screen printing. Then we moved on to inkjet printing with a research grade inkjet. And then we do dot printing, where we jet less than 200 microliters of each reagents, and we can produce hundreds of sensors in this way. And more recently, we are looking at 3D printing. So we do a lot of ink characterization and how we can maintain these bioreagents, maintain the viscosity, and the rheology of this space when designing these sensors. Our main platform is what we call NanoSerac, where we integrate serum oxide nanoparticles that have redox properties, and they also change color in reaction with the different chemicals. So, and we use these particles in lieu of organic dyes. They also function as an oxidase enzyme catalyst, so we are getting a significant enhancement for reaction with oxidase-like enzymes. And selectivity is achieved through enzymes and aptamer type chemistry. Just to give you an idea of how this looks compared to colorimetric assays. So I just use here an example of glucose. It applies to every single glucose, every single oxidase enzymes. If you want to do a colorimetric test and you buy a test from Sigma, it's about $200, $300. You can use it within a certain amount of time. It has 3, 4 reagents that need to be mixed in a certain way. where essentially this glucose oxidase in this case, and then peroxidase, and then an organic dye, all of this to detect the enzymatically generated hydrogen peroxide. So we replace both the dye and the peroxidase enzyme with inorganic, very stable serum oxide nanoparticles, and the color changes instantaneously almost from incolor to dark yellow, and very low cost, and everything in our assay is immobilized and printed onto a piece of paper or cotton. So based on this concept, relatively straightforward concept, we now have sensors for hypoxanthine, lactate, glutamate, glucose, biogenic amines, and ethanols by simply changing the enzyme. We are using the same immobilization chemistry, silanization with chitosan and alginate in between. And these are examples of for glucose, but the same kind of examples we obtain for lactate, glutamate, and other food compounds. We can also do detection of pesticides by inhibiting enzyme cholinesterase sensors. So we go from non-toxic to neurotoxic if the color disappears, and we measure this, all these colors you can measure with a color meter. We can also measure it with a cell phone. These are the same type of devices that people use that when they design paint. So every single color has a unique code that we can relate with composition and we can relate with concentration. One of my students was very interested in monitoring antioxidants in food and assigning a code capture with this color capture device to each type of green tea or mushrooms. She tested wine, she tested cosmetic products. The sensors are extremely easy to use. We can sell them actually for people who are interested in authentication or non-antioxidant compounds. These are some examples from tea, different types of teas, and the value, the intensity of the color, it basically relates with the antioxidant capacity in the sample. Measurements is simply dipping this modified paper sensor into the tea. For mushrooms, we do need to do an extraction step, so we can't really put it on top of the samples. One potentiality for this type of sensors is also for food and product authentication, maybe for uses of raw to authenticate composition or changes on composition of raw materials. These are examples for wine and cosmetics. At the time of purchase, we can use these sensors to measure the degradation over time after a couple of months storage. So we can have an actual measure of the antioxidant content at the time and of use. We further expanded this chemistry to measure the products of ATP degradation, which happens in fish and meat, and design a sensor for hypoxanthine by using the serum chemistry and xanthine oxidase. We started with our nanoserac assay, and we quickly realized that the sensitivity of the assay, the yellow changes, are not as strong, especially for large-scale use. And we then changed the chemistry with the formazan compound. So we mobilize on the paper and xanthine oxidase enzyme, which reacts, and the formazan and NBT dye, which reacts with the hydrogen peroxide and forms this beautiful, very strong precipitate with dark purple. So we can produce them in the same way. And the color changes as we look at the purple color is significant. The amount of hypoxanthine that we measure is exactly the amount released. It matches the concentration released during the food degradation process. So my student tested this sensor. She's essentially put it on top of the fish with no sample treatment and let it test it after a couple of hours of degradation at room temperature, after 4 hours, 24 hours. And we can assign a freshman indicated based on the color change. So we can envision something that is fresh, that has no color or very faded color. And then as the amount of hypoxanthine increases, then the concentration of the hypoxanthine, it's inducing increase in the formazan production. And that's why we see this very dark purple color. Other sensors that we've developed are for bisphenol A. Here we use a slightly different chemistry. We are using polyphenol oxidase enzyme, which is very stable. Actually, at room temperature, it can degrade within a week, but by immobilizing within this layer by layer sequence of chitosan and alginate, these sensors are stable for about a year at room temperature, so which was completely unexpected. And that's something that we show that by carefully designing the surface chemistry, we can increase the stability of biological reagents. So this is a bisphenol A. We used it in urine, in indoor air, and in all sorts of samples, and the reaction is very, very robust. We are now doing these experiments in the biochemistry class with students, so you can get something that is more robust than this method in terms of paper sensors. Another sensors we've done is for heavy metal detection. This is a copper sensor that we printed first with dot printing, and now we do a lot of 3D printing for quantitation. We used it on copper octoide or copper sulfate, which are the most used pesticides in agriculture. And we try to measure the exposure to copper on leaves, directly on leaves. So we have a piece of paper or a dot or this plant tool. So for example, that we place this on top of fruits and vegetables. So where we are going next with this, my students are doing a lot of work in 3D printing and manufacturing, not only the sensor, but actually the entire film. And we try to impart the color changing ability by incorporating different dyes or materials that are photochromic or electrochromic. So these are just some examples of a copper sensor. This is a flexible 3D printed packaging type material that is based on nanocellulose that could potentially be used for food packaging. And one last example, we do a lot of electrochemistry work as well, not that much for food packaging, because you still need to use the electrochemical system and all the connectivity with it. And we are very excited, and that's the project we are working. One of my students is working is a low-cost electrochemical sensor for PFAS, which has drawn a lot of attention in the field. So we measure now with electrochemical means PFAS at picomolar levels, and there is evidence of the presence of these contaminants in the food. This is a field that is completely unexplored, and I think we'll see a lot of developments in the future. So with that, the next generation, one can envision connecting the sensing technology, either electrochemical or colorimetric devices with the Internet of Things, and develop truly field deployable sensor or sensor arrays, send it to a cell phone or a centralized computer system, transmits the data, and inform the community, industry, or regulation on status of food. Before we get there, I think we have a number of technical limitations that people who are in the sensing field should be aware of, that should go beyond the excitement of publishing papers, but maybe spending a little bit more time on improving the sensitivity, really testing real samples, move away from only doing standard samples in the lab, because it's very different. Everything, as soon as we translate the sensor sensor into real samples, we have all sorts of cross-contamination or passivation of surfaces. Stability continues to be a problem. Sample variability is huge, particularly in the food area. Miniaturization is important, and sensor connectivity, it becomes important as you use this large number of samples. Regulation and standardization, and I will add here, really, it's a very multidisciplinary field. I think we are a sensor group and analytical chemistry group. I am in an insular position here. We don't have a food chemistry program, so we design sensors up to a point, and I think the next step will be really to test this and see the capabilities on real samples in real situations, or even do we need these sensors even? What are the most important analyses that people in the sensing field should look for? And all of these require interdisciplinarity. We can do Internet of Things without a computer expert. We can't design the surfaces without materials experts. We can do manufacturing or printing without advanced manufacturing experts. So I think the message is the collaboration between academic industry to push innovation and discovery beyond academic publications to really facilitate the transition of this technology. I think there is a lot of potential, many of the sensors and all the sensors that I showed you today are very reproducible. We can make them in hundreds. We can, they are very easy to use. They are all reagentsless. Everything is integrated within, we need to demonstrate functionality in real world conditions and do a lot of real sample analysis. So with that, I will end here, and I'm happy to answer any questions. Thank you.
Thank you very much. Yeah, very nice talk.